So, Dr. Woeller here for Autism Recovery System. I want to welcome everybody to this webinar. Probably got about 30, maybe 35 slides, so this won't be too long of a presentation. Important topic. I wanted to have this conversation because there is a lot of labs in the integrative medicine space that are reporting a specific chemical called DHPPA as something significant for Clostridia. And there's a lot of confusion around it. So we're really going to emphasize HPHPA, which is a very important compound, and we'll kind of evolve this conversation. So the topic here is autism and the importance of organic acid testing of the Clostridia bacterial marker, HPHPA. So when we know within the medical world with regards to autism, there's a lot of things happening: digestive problems, nutritional imbalances, methylation issues. And we certainly could say that Clostridia bacteria are part of that equation for some kids, not all, but many. And one of the things that I developed and sort of have worked off of now for many years is something called the four-pillar approach to biomedical intervention. And this really comes down to the fact that we know we should start with diet, and there's not one autism diet. It's just a real food, whole food diet that has to be nuanced for a particular child. But fundamentally, it's important because it's not only a nutritious diet, but it also helps with improvement of health within the digestive system. And then we know also that many kids have nutritional deficiencies, either because of poor diet or they're very selective with foods, maybe poor absorption from the gut. And so they're often nutrient deficient in a wide variety of things. And then, of course, the digestive system plays a huge role in some of the issues that are going on because we know that the gut and the brain communicate. And then, of course, the methylation system is often impaired in these kids. But we're going to focus on the gut and just kind of focus specifically on Clostridia. So, Clostridia bacteria is something that all of us are exposed to. These are things in our environment as well as our own body. And the characteristics of Clostridia bacteria in general is they're called obligate anaerobes, which means they do not like oxygen. Now, they can live outside the body, and they do so through a form called a spore. So it's sort of like, think of the spore like a coconut, essentially. It's got a really hard shell. And then it gets into our body and breaks down and it can become an infection. But we also have Clostridia bacteria that are a normal part of our microbiome as well. So many of the Clostridia bacteria are non-pathogenic, but there are some members of the Clostridia family, essentially, that are extremely problematic. And some of these produce very toxic compounds. So Clostridium tetanus, for example, or Clostridia botulinum are examples of Clostridium bacteria. So there's a number of disease causing Clostridia. Most of us think about Clostridium difficile as the one that is causing most problems. But again, these things are abundant within our digestive tract. A large percentage of our microbiome is made up of Clostridia bacteria, but not all of them are pathogenic. But Clostridium botulinum, for example, causes botulism. So this can cause paralysis, for example, and can be lethal. Clostridium perfringens is a common cause of food poisoning that can lead to cellulitis and, in really severe cases, gangrene. We've got Clostridium tetani, which we know can cause tetanus, and Clostridium difficile, which in some individuals can lead to inflammatory bowel disease and a very serious condition called pseudomembranous colitis. Clostridium histolyticum, Clostridium sordelli, these are also involved in gangrene and also infections sometimes post-abortion. Clostridium sordelli is probably not something you would see all that common. It's fairly rare. But again, most of us have Clostridia bacteria that are, again, not lethal, but they're causing some type of problem. And this is where the organic acids test comes into play. So most of the time when we're trying to evaluate for Clostridium difficile, we're testing for two specific toxins, toxin A and B, which are the main virulence factors of Clostridia difficile in the digestive system. However, other Clostridia, as well as other strains of Clostridia difficile, can produce different compounds that don't really have their negative effect in the gut, but they can adversely affect the brain and nervous system. And that is something like the HPHPA and the 4-creosol. Now, if you've ever done an organic acid test on your child, I have worked with Great Plains Laboratory for years in the past. Great Plains became Mosaic Diagnostics. This is still my preferred organic acid test. And we can see that the previous format is on the right. The new format of the organic acid test is on the left. This is the page 1 of the intestinal microbial overgrowth chemicals. And again, HPHPA is something that shows up fairly commonly as the main Clostridia toxin that the organic acid test picks up on. So again, it's not just a Clostridia problem. There's food sensitivities, there's environmental toxins, there's nutrient deficiencies, for example, but all of this can make up the puzzle scenario of any given individual with autism and how it might compromise them. Now, one of the things that I've seen clinically over the years in the presence of Clostridia bacteria is sometimes behavioral problems, particularly irritability, moodiness, agitation, and which can sometimes spill over into aggression or self-injurious behavior. I don't want to make anybody feel that somehow this is the only cause of those problems. But if that's happening, then your child should be assessed for the presence of Clostridia bacteria through the organic acid test, specifically looking at HPHPA and 4-creosol. One of the problems of Clostridia bacteria in general, specifically, or most commonly, I guess you would say, is the Clostridium difficile, is that there is a wide amount of people who have recurrent problems. So it's not something that's easily treated. It tends to come back. We see the same pattern with many of the kids where they just don't clear, or it's not a lasting effect to have the Clostridium toxins eliminated. So there's nuances to this. It could be other microbiome imbalances. It could be other exposures that might be happening that are weakening the immune system. So a number of factors, but one of those that commonly contributes to the ongoing problem of microbiome disruption in the gut is glyphosate exposure. And so glyphosate, what it's doing is that it inhibits the microbiome in various ways. Now, glyphosate is something that is sprayed on GMO foods. So, but what's interesting about glyphosate is it specifically inhibits an enzyme called EPSP synthase. And EPSP synthase inhibits plants from making a specific chemical, this very long chemical right here, that helps in the utilization of tyrosine, phenylalanine, and tryptophan, these amino acids, for plant metabolism. Well, the problem is that within our microbiome, we also have bacteria, and that bacteria gets inhibited by EPSP synthase from the glyphosate, or inhibits it, and that causes the bacteria to lose their ability to utilize these amino acids. And that creates a microbiome disruption. In fact, many of the animal studies where they've looked at glyphosate exposure in the microbiome, it causes a decrease in the ratio of beneficial to harmful bacteria. So what does that mean? Well, we can get an overgrowth or an expression of pathogenic bacteria like Salmonella and various Clostridium bacteria. At the same time, we're getting a depletion of normal healthy bacteria, like a decrease of Enterococcus or Lactobacillus or Bifidobacterium. And that's a problem because things like Bifidobacterium produce many compounds like butyric acid, which is needed as a fuel source for the digestive system, including the mitochondria of the body and the mitochondria that service the cells of the digestive tract. So there's a lot of downstream effects that can happen with things like glutathione exposure. Not glutathione, excuse me. um, glyphosate exposure. So let's focus a little bit more on HPHPA and 4-creosol. So when we look at an organic acid test coming from Mosaic Diagnostics, about 1 in 4, 1 in 5 organic acid tests will have at least one elevation of a Clostridia marker. The most common marker that we see high is HPHPA. Shows up at about 80% of the time. And then we've got 4-creosol, which is also shows up fairly well, not as high, about 15, 20% of the time. So the 4-creosol discussion is important because it's known that Clostridia difficile, at least certain strains of Clostridia difficile, can produce 4-creosol. So P-creosol is the same thing as 4-creosol. And this particular compound will inhibit other types of competing Clostridia bacteria in the digestive system. So the problem is that in the presence of 4-creosol, we can inhibit an enzyme called dopamine beta hydroxylase. And why is that important? Because dopamine beta hydroxylase is what converts dopamine to norepinephrine. And this is important for energy production in the body, all throughout the body. And when we inhibit that enzyme, we can start to get a buildup of dopamine metabolites, other chemicals. And these themselves have some toxic effects, and that can be problematic. So it can cause other problems in the brain and nervous system. Now, HPHPA, as I mentioned, this is the one that shows up most commonly. And so it's the one that you're going to see most commonly on the organic acid test, but this is the one that also causes a lot of confusion because its acronym sounds or looks similar to DHPPA. So what is HPHPA? Well, it stands for this particular chemical, this 3,3-hydroxyphenyl 3-hydroxypropionic acid. And it's, basically that's the organic chemical or chemistry name of it. It has a propionic acid that is attached at carbon 1. And that's a problem because propionic acid at very high levels can somewhat interfere with the Krebs cycle, which is important in our mitochondria for energy production. Well, Dr. Shaw, Bill Shaw, who started Great Plains Laboratory many years ago, actually found that HPHPA was much higher in autistic children than neurotypical children. It wasn't that neurotypical children couldn't have HPHPA. It was just that the HPHPA was found to a much higher degree in children, teenagers, well, in this case, particularly children with autism. So it's a fairly common marker. Well, what can happen? Well, HPHPA and 4-creosol are both known to inhibit this dopamine hydroxylase enzyme, which is what's needed to convert dopamine to norepinephrine. If we get enough of that inhibition, we start to get a buildup of dopamine, which has some toxic influences in the brain and nervous system. And we get some decrease of norepinephrine and epinephrine. So this particular image up here kind of shows what are some of the things that are related to norepinephrine. So it helps with alertness, concentration, and energy. It also, when it's imbalanced, can contribute to things like anxiety or irritability. Dopamine is important for reward and motivation, but when it's imbalanced, it can lead to aggression, for example, and moodiness. So there's individual activity of these particular neurochemicals, and then there's overlapping activity. What we commonly see with kids when these markers are high is often aggression, cognitive problems, moodiness, alertness, concentration issues, irritability, impulsivity, for example. So one of the other adverse influences of too much dopamine in the brain and nervous system is that dopamine is very reactive. So it can end up squelching a lot of the glutathione that we need. And glutathione is an important antioxidant, helps to protect the nerve cells, helps protect the brain. We need it for detoxification purposes. And again, this unregulated dopamine, because of a buildup, leads to a lot of internal stress within the nerve cells. And one of the chemicals that gets produced is something called dopamine O-quinone. And dopamine quinone can actually damage nerve cells to where the nerve cells sort of prematurely eliminate themselves from their community. It can also generate a lot of reactive oxygen species, which damage mitochondria. Long-term implications of excess dopamine can lead to neurodegeneration, which this could be something seen more like with Parkinson's disease, for example. However, in kids, the same underlying mechanism can be at play. That's maybe just creating some neurodevelopmental challenges. It could be leading to behavioral challenges, etc. So what is the difference then between DHPPA and HPHPA? Because again, there are some labs that are reporting DHPPA, but not HPHPA. And there's a very important distinction. When you look at the organic acid test from Mosaic Diagnostics, under the bacterial section, there are 5 markers: hyperic, 2-hydroxyphenylacetic, these other 2, and then DHPPA. But it's important to understand that DHPPA is listed as a marker of beneficial bacteria. Now, notice in this particular case, it's elevated. What does that mean? Okay, what does it mean when DHPPA is elevated? Well, DHPPA has a very similar sounding name to HPHPA, but it gets produced by many normal healthy bacteria in the digestive tract, like lactobacillus and bifidobacteria. There is a Clostridium bacteria that produces it, but it's typically found at very, very low amounts. Most of the time when we see DHPPA elevated, it's because somebody is eating a number of different foods. And this is just a short list: parsley, thyme, celery, for example. It can also be elevated in the presence of probiotics. I've actually had kids who were put on antibiotics because this marker was high. And to me, that's a problem. So when you actually look at an organic acid test like this, we can see we've got some general bacterial markers of dysbiosis, and we have a DHPPA. But what marker is most concerning? Well, what's most concerning from this list is HPHPA, for the reasons I just mentioned. It creates stress within the neurotransmitter system of the brain. It could be something that could cause or contribute to moodiness, irritability, impulsivity, other behavioral problems, whereas we don't see that with DHPPA. And so again, this is the most concerning marker. But this often gets overlooked, particularly if there's a lab that's not reporting HPHPA. So the question is, in this particular example, is this a cause for concern? Is this elevation of DHPPA meaningful from a clinical problematic standpoint? Look at our Clostridia markers down below. Okay, they're all completely normal. Our DHPPA is present. Yeah, it's slightly high, right? But is it possible that this is just coming from diet or just increased activity of normal bifidobacterium or lactobacillus, for example? Perhaps it's coming from a probiotic. Would you actually treat based off of this marker? In my experience, no, you would not. It would be inappropriate to actually treat with, some type of antimicrobial compound for just DHPPA being elevated. So PubChem is a website that's overseen by the National Library of Medicine from the National Institutes of Health. And DHPPA is actually listed and referenced as having antioxidant capacity. at reducing pro-inflammatory cytokines. So cytokines are immune chemicals that drive inflammation. There may even be some protective mechanisms of DHPPA against pathogenic bacteria. In fact, a relatively recent article came out, this was 2022, called the gut microbial metabolite 3,4-dihydroxyphenylpropionic acid, which is DHPPA, alleviates hepatic, which is liver ischemia, and mitigates macrophage pro-inflammatory activity. So what does that mean? Well, what they were looking at, this was a mouse study, was that when there is liver ischemia, sometimes when the liver reperfuses with the blood supply, that event can actually trigger widespread inflammation in the heart. So there has to be kind of a regulated return of normal blood flow. If it comes in too quickly, too robustly, it can trigger inflammation and be problematic. This is what's called reperfusion injuries. Well, it turns out that what they found was that when there is an abundance of good bacteria in the digestive system, and there can be a production of DHPPA, right? This is the marker that the organic acid test references being linked to beneficial bacteria. It can actually inhibit a chemical sequence within these macrophages to decrease its pro-inflammatory activity, decrease the liver's injury from that reperfusion scenario. So the bottom line is that DHPPA is actually having a positive influence outside the gut, in this particular case, at regulating runaway inflammation. So you're not going to see that with HPHPA. So as we wrap this up, the main point is when you are evaluating your child's organic acid test, or you're working with a doctor who's looking to do an organic acid test, make sure you pay attention to the HPHPA if it's present on the OAT. It should be. If it's not, then it's not a test I recommend doing. Also pay attention to the DHPPA, because again, remember the DHPPA is linked to normal bacteria. The HPHPA is linked to pathogenic bacteria. So within the Autism Recovery System website, we have a lot of information. You can access and post questions to me through the member forum. There is also a biomedical course that is available along with videos and articles. This Autism Biomed Essentials course is 6 modules, probably 8+ hours, give or take, of educational material, a lot of downloads, PDF downloads, for example. So a lot of general resources, a great course if you are new to the world of biomedical intervention or want to learn a little bit more. So all of that is available to you through membership of the Autism Recovery System website. Okay, so that was it. That's the information I wanted to present. Appreciate everybody's attention. And again, I'm Dr. Kurt Woeller for Autism Recovery System. We'll see you next time. Thank you.